Okay, so I will introduce as quickly as I can the open science services we have built or gathered in PHENET to deal with data management, integration, sharing, and modeling. So in PHENET, the data management is dealing with phenotyping data, environment, a little bit of microbiome for some soil use cases where they are doing metabarcoding. And the objectives are first to enable data flow from the acquisitions to the modeling. Therefore, those workflows will rely on services, data standard, tools. We want also to ensure sustainable data publication standardization from experimentation to modeling and integration, and of course, ensure this beyond the end of PHENET. We want also to provide services to enable fair data curation, storage, and exchange. And finally, connect to European data portals to ensure the visibility of PHENET results, either through the EOS data portal or through FEDER. For this, we are currently using existing solutions from Elixir, NI, or Emphasis. So the data lifecycle for phenotyping and beyond, not only phenotyping. So it's a classical one we find in life science with first the planning of the experimentation, collecting of the data, processing of those raw data to aggregate them, consolidate it in a form that enables analysis, preservation of the result, enabling sharing and future reuse. Of course, it is not always as simple as it looks, because in our case, and in most of yours probably, it's not about a single experiment, but about many experiments, either in greenhouse plus a phenotyping network, plus an automated network with a lot of sensors. And all of those results, all of those data needs to be aggregated, integrated to enable, once again, analysis and preservation. So we have a set of services for data exchange and workflow. I'm going to focus today on platform information systems. I will not speak too much about portable devices or sensors, neither of scripts or workflow or data repositories or data portals. I will also speak about global data standards and the one we have decided to use. So the plant data standards, the first one are about structuring, formatting, organizing the data set description, in other words, the metadata that are used to correctly identify the experiments. It allows to build data models, it uses data templates, it includes a description of phenotyping variable on biological material. And among those type of data standards, you can find VCF and GFF for genetic data, MyAPI and ICASA for phenotyping. And the last thing I would like to underline is that those data standards are built and maintained mainly through the collaboration between biologists and computer scientists. So in PHENET, there are two data standards that we are interested in. First, MyAPI, minimum information about plant phenotyping experiment. It addresses the needs of crops and woody plants in single experiment and multi-local, multi-year network. In field or greenhouses. It has been built by many people all around the world through an open community, and it is still very active. ICASA, the International Consortium for Agricultural System Applications, it deals with phenotyping and agronomic data, especially with a focus on interoperability toward modeling softwares and systems. It has been built within AgMIP using the ACE data schema and data dictionaries based on published ICASA standards. So those standards are specifications that need to be implemented. For the example of MyAPI, there are several databases and data repositories that implement it. In particular, FAIRS that I'm going to present a little bit later. In fact, any breeding API compliant database. as well as generic data repositories such as Dataverse and Zenodo that can implement MyAPI by simply reusing the MyAPI data template. This template used for file exchange is used not only in data repositories but also in project workflows. There are also data archives with EtherTab and AeroCrate. There is a web service implementation, so the breeding API, which is also an international collaboration. It's the standard open web service API, with the main target being breeding, but breeding at large, it goes beyond. Built by, once again, a lot of people experience for this. The CGIA have been super active in this, and the current coordinator is Peter Selby from Cornell. And finally, there is an ontology formalizing MyAPI. So this overview of what is the MyAPI data standard. So there is an investigation, a data set that holds all of the data, all of those data covering one-to-many locations during one-to-many years. Each location, each year is a study or experiment in one location during one-to-many years, in which we are holding several plant measures, data sets of file at different level, either plant or microplot, and on which we can hold trait indices, phenotype, environment, whatever type of data that makes sense, using a triplet trait method scale borrowed from the crop ontology to correctly describe the variables. ECASA is not only a structure data standards like MyAPI, but also includes semantics, and I'll get a little bit in details on what means semantics right after this. The important thing to keep in mind is that it also, like MyAPI, describe a phenotyping experiment, including and maybe with a focus on cultural practices for crop models and simulation. There is an ECASA Excel sheet which is complete, detailed. We see here the data model, in fact, the set of sheets that you can find in an ECASA Excel sheet. It complies to agronomic standards. It is widely used, and there are a lot of libraries that allow translation from this sheet towards model. So I've talked to you about semantics. Semantic is really about describing the data. It's in fact a list of control vocabularies, simple term, name, and definition. And a term can be something, any trait, for instance, plant height, or leaf area index, and so on. It therefore allows to handle phenotyping and environment variables using, once again, the trait method scale triplet. And it can be a little bit more powerful than flat list using ontologies that enable semantic linking between the terms. The important thing to keep in mind is that those control vocabulary must be biologist driven. It must not be only a computer scientist tool. Finally, there is the technical implementation that I've talked to you about to enable data integration and sharing between tools and systems. Finally, there is a question of persistent unique identifier. Especially for the biological material, we must be sure to identify what has been planted in a way that is unambiguous at the World Wide Web scale. For this, there is the MCPD standards and other standards to correctly identify cultivar and accessions. So in PHENET, we are merging ECASA and MyAPI. ECASA for the cultural practices and MyAPI for the extended plant material identification and description. And therefore, there is a formalized data modelization that is currently in progress, that is being gradually loaded into the phase information system to enable a workflow where those data will be easily browsable, visualizable in fields, where we would have export mechanism toward data publication to data repositories and modeling softwares. Within semantics, there is also the question of data standards for soil use cases with a synergy between MyAPI and Glosys, and this will be fully presented during the last webinar of this series. Finally, I would like to underline the phenotype experiment data management services in PHENET. So first, there is PHENotyping. It's a dedicated information system, specially designed for instrumented and complex experiment where we measure a lot of different level, plant, microplot, whole field, with a lot of high throughput sensors and vectors and systems. It allows organizing the dataset for retrieving the info. This system is very well suited with complex experiments, but for simpler use case, we need also something that is easier to deploy. Therefore, there are workflows based on file templates, such as MyAPI, for simpler dataset where there is little to no sensor, for instance, to handle phenology on field. There is also a file validator and an example of such workflows described in this DOI. This is not fully considered for PHENET. So far, there is little to no direct use for that, or at least for that workflow as we have seen it. As we will see, the intercropping use case is using the file approach. So in the GBIA and plant health use cases, and for instance here the Cyril use case, we have the use of PHENotyping, which has been well established in phenomorphosis for years now, where we gather environmental and phenotypic data with REST API web applications and this dedicated database, which provide both a web user interface for easy browsing and the breeding API to enable automatic data access. There is also a connection with a file system based on iRoads that allows to handle images with thousands of files, hundreds of terabytes of data. And this is connected to the sensor, phenomobiles, drones generating the images and hold them before they are being processed for trait and index extraction. Those resulting indices extractions are being stored in fields, and fields also allows a direct access to the images. For the ORCARD, there is currently an evaluation in progress deployed on the EGI infrastructure with environmental data and phenotypic data once again, but no image processing or trait in this extraction directly handled using iRoads. They are using whatever is available in their use case, but not the services built within PHENET and the partners infrastructures. The rest of the PHES system remains exactly the same. So the PHES user interface allows management of the scientific organization with project information, facilities, sensors. The variables, the list of scientific objects, so what we have done the experiment on, the germplasm, identification of the biological material. And also very important because there is this processing of the data, the traceability of the sensor being used, the raw data processing software and parameters, the link to data visualization. So in general, a need of provenance that can be used either through PHES or through companion file in most cases, where we have the trace of how multiple variables can have been processed into indices combining those variables. So as a perspective, the things that I'm not going to describe today, so there are the guidelines that help people to understand how to manage their data and the data portals to give the visibility to the data. All of this work has been done by, as you can see, a lot of people. And I am done.